Hello there, and welcome to my channel. I'm so glad you found your way here tonight. Whether you're tucked under warm blankets, curled up on a couch with a cup of tea, or simply looking to unwind after a long day of being wonderfully complicated human, tonight we're going to take a gentle journey into one of the most extraordinary stories science has ever written, the story of CRISPR, the molecular scissors that can edit the very code of life itself. Before we begin, I'd love to know where in the world you're listening from and what time it is in your corner of the night. Are you drifting towards sleep in Dublin at 11:30? Or perhaps it's the quiet hours of a Sunday morning in Sydney? There's something deeply comforting about knowing this little corner of curiosity reaches sleepy souls across time zones, all of us wondering together about the mysteries tucked away in our cells. If you find these gentle explorations through science soothing, I'd be ever so grateful if you tap the like button and perhaps subscribed for more peaceful journeys into the wonders of our world. It helps other curious minds find their way here, and it means I can keep crafting these soft scientific lullabies for you. Now, I do hope this isn't so fascinating that it keeps you awake though. If it does, at least you'll drift off knowing something remarkable about the elegant machinery hidden inside every cell of your body. So settle in, let your thoughts slow down, and let's begin this quiet adventure into the book of life. Picture this. Somewhere in your body right now, in a cell so small you'd need to line up about 40,000 of them to span the width of a human hair, there's a library. Not the kind with wooden shelves and hushed whispers, but a library written in a language far older and more fundamental than any human tongue. It's written in just 4 letters, A, T, G, and C, and it contains the instructions for building and maintaining you. This is your genome, your personal copy of the book of life. Every chapter, every paragraph, every punctuation mark has been carefully preserved and passed down through billions of years of evolution. It's the recipe for your eyes, the blueprint for your heartbeat, the manual for how your neurons fire when you laugh, or remember the taste of your grandmother's cooking. It's elegant, it's ancient, and until very recently, it was essentially unchangeable. But what if I told you that we now have something like a word processor for this book? Something that can find any sentence, any word, even any single letter, and change it with the precision of a master editor? What if we could correct the typos that cause disease? Add new chapters that grant resistance to illness, or even rewrite entire sections to help our bodies adapt to a changing world. This isn't science fiction. This is CRISPR, and it's quietly revolutionizing everything we thought we knew about what it means to be human. The story begins, as many great scientific tales do, with curiosity about something seemingly mundane. In the late 1982nd, scientists studying bacteria noticed something peculiar in their genetic code. Scattered throughout the bacterial genome were strange, repetitive sequences, the same short stretch of DNA letters repeated over and over, like a molecular stutter. They called these sequences clustered regularly, interspaced short palindromic repeats, which thankfully was shortened to the much more manageable acronym CRISPR. For years, these sequences were a mystery. They sat there in the bacterial genome like ancient hieroglyphics waiting for someone to crack the code. Some scientists suspected they might be junk DNA evolutionary leftovers with no real purpose. Others wondered if they might be involved in bacterial reproduction or stress response. But the truth, when it finally emerged, was far more elegant and surprising than anyone had imagined. These repetitive sequences, it turned out, were part of the bacterial immune system. But not just any immune system. They were part of a sophisticated biological memory bank that could recognize and defend against viral invaders. You see, bacteria face a constant threat from viruses called bacteriophages, tiny molecular pirates that inject their genetic code into bacterial cells and hijack the cellular machinery to make copies of themselves. But bacteria, despite their simplicity, aren't defenseless. When a bacteriophage attacks, some bacteria can capture a small piece of the invader's DNA and insert it into their own genome, right between those repetitive CRISPR sequences. It's like taking a mugshot of the criminal and filing it away in a genetic police database. Then, if that same virus ever shows up again, the bacteria can recognize it immediately and deploy a molecular bouncer to escort it out of the cell permanently. The bouncer in question is a protein with the rather intimidating name of Cas9, which stands for CRISPR-associated protein 9. Think of Cas9 as a pair of molecular scissors, but scissors with a very particular talent. They don't just cut randomly, they can be programmed to cut at exactly the right spot, guided by a small piece of RNA that acts like a GPS system for the genome. Here's where it gets beautiful. The bacterial cell creates a guide RNA that matches the sequence of the invading virus. This guide RNA then teams up with the Cas9 protein, and together they scan through the cell's contents, looking for that exact genetic sequence. When they find its SNP, the viral DNA is cut, rendering the virus harmless. It's like having a molecular search and destroy mission that can find one specific sentence in a library of millions of books and remove it with surgical precision. For billions of years, this system worked quietly in bacteria, an elegant solution to an ancient problem. But then, in the early 2010, scientists began to realize something extraordinary. If this system could be programmed to cut specific sequences in bacterial cells, could it be reprogrammed to cut specific sequences in any cell? Could we borrow this bacterial immune system and turn it into a general-purpose genome editor? The answer, as we now know, was a resounding yes. And that realization changed everything. The breakthrough came when researchers figured out how to create custom-guide RNA's molecular instructions that could direct the Cas9 scissors to cut at any desired location in any genome. Suddenly, scientists had a tool that could edit genetic code with unprecedented precision. Need to fix a mutation that causes sickle-cell disease? CRISPR could do that. Want to engineer crops that can survive drought or resist pests? CRISPR could do that too. Hoping to create better models of human disease for research? CRISPR made it possible. It was as if we'd been trying to edit books by tearing out entire chapters, and suddenly, someone handed us a word processor that could change individual letters without disturbing anything else on the page. The precision was breathtaking, the possibilities seemingly endless. But with great power, as they say, comes great responsibility. And CRISPR represents perhaps the greatest power humans have ever wielded over the fundamental machinery of life itself. We're not just observers of evolution anymore, we're active participants, editors of the very code that defines what it means to be alive. This is both thrilling and terrifying. On one hand, CRISPR offers the promise of curing genetic diseases that have plagued humanity for millennia. Imagine a world without Huntington's disease, without cystic fibrosis, without sickle-cell anemia. Imagine crops that could feed billions while using less water and fewer pesticides. Imagine livestock that are naturally resistant to diseases that currently require antibiotics. On the other hand, we're venturing into uncharted ethical territory. If we can edit the genes of human embryos, should we? If we can enhance intelligence or physical capabilities, who gets access to these improvements? If we can make changes that are passed down to future generations, how do we ensure we're not making mistakes that echo through centuries? These questions don't have easy answers, and they're questions that every one of us will need to grapple with as CRISPR technology becomes more powerful and more accessible. Because make no mistake, this isn't just a tool for scientists in white coats. This is a technology that will touch every aspect of human life, from the food we eat to the medicines we take to the very genes we pass on to our children. But tonight, as you rest your head and let your thoughts wander, you don't need to solve these enormous questions. You don't need to become a bioethicist or a molecular biologist or a policymaker. You just need to appreciate the quiet miracle that's happening in every cell of your body right now, the ancient dance of DNA and protein, the elegant machinery that keeps you breathing and thinking and dreaming. And perhaps you can marvel, as I do, at the beautiful irony that we discovered this revolutionary technology not by trying to engineer life from scratch, but by paying attention to how bacteria, some of the simplest life forms on Earth, have been solving problems for billions of years. Sometimes the most profound discoveries come not from looking ahead, but from looking around us with fresh eyes and asking, what can we learn from this? In the bacterial world, CRISPR is a story of memory and defense, of adaptation and survival. In our world, it's becoming a story of possibility and responsibility, of hope and caution. It's a reminder that life, at its most fundamental level, is information that can be read, understood, and yes, carefully edited. The bacterial immune system that inspired CRISPR has been operating for billions of years, silently protecting microscopic organisms from viral threats we never knew existed. And now, in laboratories around the world, scientists are using that same system to write new chapters in the book of life. They're correcting genetic typos that cause blindness, engineering immune cells that can hunt down cancer, and even bringing back species that vanished from the earth decades ago. The elegance of CRISPR lies not just in what it can do, but in how naturally it arose from the primordial soup of bacterial survival. Think about it. For nearly 4 billion years, these microscopic organisms have been locked in an evolutionary arms race with viruses, developing increasingly sophisticated ways to remember their enemies and fight them off. And we, in our towering complexity, have learned to harness this ancient wisdom. It's rather like discovering that your grandmother's simple recipe for healing tea actually contains compounds that modern medicine is just beginning to understand. The wisdom was always there, waiting patiently for us to notice it, to appreciate it, and to learn from it. But let's step back for a moment and consider what this really means for you, lying there in the gentle darkness, your body quietly maintaining itself while your conscious mind drifts toward rest. Every 7 seconds, somewhere in your body, a cell divides. Every division requires your entire genetic code, all 3 billion letters of it, to be copied with extraordinary precision. Most of the time, this happens flawlessly. But occasionally, just occasionally, there's a mistake. Usually, these mistakes don't matter. They might occur in a stretch of DNA that doesn't code for anything important, like a typo in the margin of a book that doesn't affect the story. But sometimes, rarely, a mistake occurs in a critical spot, a single letter changed in a gene that's essential for proper function. And that tiny error, smaller than the smallest thing you can imagine, can cascade into disease, disability, or death. This is where CRISPR enters our personal stories. For the first time in human history, we have a tool that might be able to go back into that vast library of genetic information and correct those errors. Not with the crude instruments of the past, chemotherapy that destroys healthy cells along with cancerous ones, or organ transplants that require a lifetime of immune suppression, but with the precision of a master calligrapher correcting a single misplaced stroke in an otherwise perfect manuscript. The first human trials of CRISPR therapy began just a few years ago, and the results have been nothing short of miraculous. Children born with severe combined immunodeficiency bubble boy disease have had their immune cells edited outside their bodies and then returned, transformed from vulnerable to vigorous. Adults with sickle-cell disease, who lived with constant pain as their misshapen red blood cells struggled to carry oxygen, have seen their symptoms disappear after a single treatment. Victoria Gray was one of those adults. For years, she endured monthly visits to the hospital as pain crises racked her body. The sickle-shaped cells that gave her disease its name would jam up in her blood vessels like traffic in a narrow tunnel, cutting off oxygen to her tissues and causing agony that would last for days. Then, in 2019, she volunteered for an experimental CRISPR treatment. Doctors removed some of her bone marrow cells, edited them to produce healthy hemoglobin, and infused them back into her body. Today, Victoria's hemoglobin levels are normal. Her pain crises have stopped. She can play with her children without fear of triggering an attack. In the language of medicine, she's been cured. But in the language of human experience, she's been given back her life. Stories like Victoria's are becoming more common, but they're still rare enough to feel like miracles. And in a sense, they are miracles, not divine interventions, but the result of decades of painstaking research, of scientists asking questions about bacterial immunity and following those questions wherever they led. Yet for every success story, there are reminders of how much we still don't understand about the intricate dance of genes and proteins that makes life possible. CRISPR, for all its precision, is still a relatively blunt instrument. It's like having a word processor that can find and replace any word in a document, but sometimes it might accidentally change words you didn't intend to modify. Scientists call these off-target effects, and they represent one of the biggest challenges facing CRISPR research today. Imagine you're trying to correct a typo in a vast library, but the library is written in a language you're still learning to read. You find the word you want to change, but there might be other instances of that same word elsewhere in the library that you don't want to alter. Or worse, there might be similar-looking words that serve completely different functions. This is the challenge of genome editing. The human genome is so large and complex that ensuring you're only changing what you intend to change requires extraordinary care. This is why current CRISPR therapies often work with cells outside the body, where scientists can test them extensively before returning them to the patient. It's why researchers spend years developing new versions of the CRISPR system that are more precise, more reliable, more predictable. And it's why every clinical trial is monitored with the kind of attention usually reserved for space launches or diplomatic negotiations. But precision isn't the only challenge. There's also the question of access. The first CRISPR therapies are extraordinarily expensive, hundreds of thousands of dollars per treatment. They require sophisticated laboratory facilities, highly trained technicians, and months of careful preparation. For now, they're available only to patients enrolled in clinical trials, or to those wealthy enough to afford experimental treatments. This raises profound questions about equity and justice. If CRISPR can cure genetic diseases, who gets to be cured? If it can enhance human capabilities, who gets to be enhanced? Will we create a world where your genetic destiny depends not just on the lottery of inheritance, but on the size of your bank account? These aren't abstract philosophical questions, they're urgent, practical concerns that societies around the world are grappling with right now. Some countries are investing heavily in making CRISPR therapies widely available through public health systems. Others are taking a more cautious approach, waiting to see how the technology develops and what unforeseen consequences might emerge. And then there are the questions that keep bioethicists awake at night. Questions about where we draw the lines around human enhancement. Most people would agree that using CRISPR to cure a child's blindness is morally justified. But what about using it to ensure that child never goes bald? Or to give them perfect pitch? Or to boost their IQ by 20 points? The line between treatment and enhancement isn't as clear as we might hope. Is correcting a genetic variant that increases the risk of heart disease a medical treatment or a form of enhancement? What about editing genes to provide resistance to HIV, or to help someone live to 120 instead of 80? As our understanding of genetics deepens, we're discovering that many of the traits we think of as normal variations, height, intelligence, athletic ability, have genetic components that could theoretically be modified. Some scientists argue that we should embrace these possibilities, that we have a moral obligation to give our children the best genetic start in life possible. Others worry that genetic enhancement could exacerbate existing inequalities or create new forms of discrimination. Still others question whether we're wise enough to make permanent changes to the human genome when we're still learning how it works. Perhaps most challenging of all are questions about heritable editing changes that would be passed down to future generations. Unlike the CRISPR therapies currently in clinical trials, which affect only the patient receiving treatment, editing human embryos could alter the genetic legacy we leave to our children and grandchildren. A single edit made today could echo through centuries of human history. In 2018, a Chinese scientist named He Jiankui announced that he had used CRISPR to edit the genes of twin girls before they were born, making them resistant to HIV infection. The announcement sent shockwaves through the scientific community. Not because the technology wasn't ready, though, many argued it wasn't, but because he had crossed a line that the international research community had agreed not to cross without much broader discussion and consensus. The twins, known only as Lulu and Nana, are now several years old and appear to be healthy, but their existence raises profound questions about consent, oversight, and the pace of scientific progress. They didn't choose to be the first gene-edited humans, yet they will carry that distinction for their entire lives. And the modifications made to their genomes, if they have children of their own, will be passed on to the next generation. The He Jiankui case illustrates both the power and the peril of CRISPR technology. On one hand, it demonstrated that the technical barriers to human gene editing are lower than many people realized. The equipment needed for CRISPR experiments costs thousands, not millions, of dollars. The techniques can be learned by graduate students, not just senior researchers. In many ways, the democratization of gene editing technology is already underway. On the other hand, the case highlighted the importance of ethical oversight and international cooperation in governing this powerful technology. Most countries now have regulations prohibiting heritable gene editing, and international scientific organizations have called for a moratorium on such experiments until broader consensus can be reached about when and how they might be appropriate. But regulation, while necessary, isn't sufficient. CRISPR technology is advancing so rapidly that regulatory frameworks struggle to keep pace. New applications emerge faster than policies can be written to govern them. And, in a globalized world, what happens when different countries have different rules about what kinds of genetic modifications are permissible? These challenges extend beyond human applications to agriculture, conservation, and basic research. CRISPR is already being used to develop crops that can grow in saltwater, survive extreme temperatures, or produce more nutritious food. Scientists are using it to create animal models of human diseases, accelerating the development of new treatments. Conservation biologists are exploring whether CRISPR could help save endangered species or even bring back extinct ones. Each application brings its own set of ethical considerations. Is it acceptable to release gene-edited mosquitoes into the wild to prevent malaria transmission? Should we use CRISPR to create livestock that feel less pain when slaughtered? Can we justify editing the genes of wild animals to help them adapt to climate change? These questions don't have simple answers, and different cultures, different religions, and different philosophical traditions may reach different conclusions. What seems obvious to one person may be morally abhorrent to another, and unlike many previous technologies, CRISPR affects the very essence of what it means to be alive, not just how we live, but how life itself is structured and passed on. This brings us to one of the most profound questions of our time. Who gets to decide? In the quiet darkness of your room tonight, as your cells continue their ancient dance of division and repair, millions of scientists, ethicists, policymakers, and citizens around the world are grappling with decisions that will shape the future of life itself. And perhaps that's as it should be. Perhaps the power to edit the book of life is too important to be left to any single group, no matter how well-intentioned. The beauty of CRISPR lies not just in its precision, but in its accessibility. Unlike previous genetic engineering techniques that required massive laboratories and decades of specialized training, CRISPR can be learned by undergraduate students and performed with equipment that fits on a laboratory bench. This democratization of genetic engineering is both thrilling and unsettling. On one hand, it means that brilliant minds in developing countries, in small universities, in startup companies, can contribute to solving humanity's greatest challenges. On the other hand, it means that the power to alter life is spreading beyond the careful oversight of major research institutions. In community laboratories across the globe, biohackers and citizen scientists are experimenting with CRISPR in ways that would have been unimaginable just a decade ago. Some are trying to engineer bacteria that can eat plastic waste or produce valuable medicines. Others are exploring whether CRISPR could treat their own genetic conditions. A few are simply curious about how this remarkable tool works, driven by the same wonder that you might feel as you drift towards sleep tonight. This grassroots enthusiasm for genetic engineering represents both the promise and the peril of our current moment. When powerful technologies become widely available, they can accelerate progress in ways that centralized research never could. The internet revolution happened not because a few large companies decided it should, but because millions of individuals found creative ways to use and improve the technology. Similarly, CRISPR's greatest contributions to human welfare may come not from major pharmaceutical companies, but from unexpected corners of the scientific community. Yet this same accessibility raises concerns about safety and oversight. When genetic engineering was confined to well-funded laboratories with rigorous safety protocols, the risks were relatively contained. But as CRISPR spreads to smaller labs, community spaces, and even home workshops, ensuring that experiments are conducted safely and ethically becomes much more challenging. The question isn't whether we can prevent the democratization of genetic engineering that ship has already sailed. The question is how we can nurture a culture of responsibility among the growing community of CRISPR practitioners. How do we ensure that enthusiasm for genetic engineering is balanced with respect for its power? How do we encourage innovation while preventing harm? Some of the most promising answers to these questions are emerging from unexpected places. In Kenya, researchers are using CRISPR to develop crops that can survive the increasingly unpredictable rainfall patterns caused by climate change. In Brazil, scientists are engineering mosquitoes that carry a gene that makes their offspring unable to survive, potentially providing a new weapon against Zika and dengue fever. In Thailand, researchers are exploring whether CRISPR could help coral reefs adapt to rising ocean temperatures. These applications share something important. They're focused on solving local problems using local expertise. Rather than waiting for solutions to be developed in wealthy countries and then exported around the world, communities are using CRISPR to address their own unique challenges. This represents a fundamental shift in how genetic engineering research is conducted and who gets to participate in it. But perhaps the most remarkable thing about CRISPR is how it's changing our relationship with our own biology. For most of human history, our genes were our destiny. The DNA we inherited from our parents determined not just our eye, color, and height, but our susceptibility to disease, our lifespan, and in many ways, the boundaries of what we could become. CRISPR is beginning to change that fundamental equation. Consider what this means for a parent whose child is diagnosed with a genetic disease. 20 years ago, such a diagnosis was often devastating a life sentence, with few treatment options and little hope for a cure. Today, that same parent might have reason for cautious optimism. Clinical trials are underway for CRISPR treatments for hundreds of genetic conditions. Gene editing might not just treat the symptoms of their child's disease, but actually correct the underlying genetic cause. This shift from fatalism to hope is profound, but it also brings new burdens. When genetic diseases were incurable, parents could focus on helping their children live the best lives possible within those constraints. Now, they must navigate a bewildering array of experimental treatments, weigh risks and benefits that are still being understood, and make decisions about their children's genetic future with incomplete information. The psychological impact of this transition is only beginning to be understood. Some families report feeling empowered by having options, even if those options are experimental and uncertain. Others describe feeling overwhelmed by the complexity of choices they never expected to face. Still others worry about what it means for their children to grow up in a world where genetic modification is possible but not yet routine. These personal struggles are playing out against a backdrop of broader social change. As CRISPR therapies move from experimental treatments to standard medical care, they're forcing us to reconsider fundamental assumptions about health, disability, and human diversity. If we can prevent genetic conditions that cause suffering, should we? If we can enhance human capabilities, what counts as enhancement versus treatment? If genetic modification becomes routine, what happens to people who choose not to participate? The deaf community has been particularly thoughtful about these questions. Many deaf individuals don't view their deafness as a disability to be cured, but as a cultural identity to be celebrated. Deaf parents sometimes want to have deaf children, just as hearing parents typically want to have hearing children. CRISPR could theoretically prevent most cases of genetic deafness, but should it? And who gets to decide, parents, doctors, or society as a whole? Similar questions arise around other conditions. People with dwarfism have rich communities and cultures built around their shared experiences. Many don't want to be cured of their dwarfism, any more than tall people want to be cured of their height. Yet CRISPR could potentially prevent most cases of genetic dwarfism. Should it be used for this purpose? What would we lose if certain forms of human diversity disappeared? These aren't abstract philosophical questions, they're urgent practical concerns that medical professionals, patients, and families are grappling with right now. As CRISPR therapies become more widely available, we'll need to develop new frameworks for thinking about genetic diversity, medical treatment, and human flourishing. The challenge is compounded by the fact that genetic traits rarely exist in isolation. The same genetic variant that causes sickle-cell disease in people who inherit 2 copies also provides resistance to malaria in people who inherit just 1 copy. The genes that increase the risk of ADHD may also contribute to creativity and entrepreneurship. The genetic factors that predispose someone to depression might also make them more empathetic and emotionally intelligent. Evolution, it turns out, is a master of trade-offs. Many of the genetic variants we might be tempted to eliminate have complex effects that we're only beginning to understand. This doesn't mean we shouldn't use CRISPR to treat serious genetic diseases. The suffering caused by conditions like Huntington's disease or cystic fibrosis is very real and devastating. But it does suggest that we should approach genetic editing with humility and caution, recognizing that our understanding of genetics is still incomplete. As you lie there in the gentle darkness, your body is performing millions of genetic calculations every second. Proteins are being made according to instructions written in your DNA. Cellular repairs are being carried out by molecular machines that have been perfected over billions of years of evolution. And your immune system is using strategies that would make the most sophisticated computer virus scanner seem primitive by comparison. All of this is happening without your conscious awareness or control. You don't have to think about making your heart beat or your lungs breathe or your neurons fire. The genetic program running in every cell of your body handles these tasks with remarkable efficiency and reliability. It's a system so elegant and complex that we're still discovering how it works, even as we develop tools to modify it. This is both the wonder and the responsibility of our current moment. We have the power to edit the genetic programs that run our bodies, but we're still learning how to read them. We can change individual letters in the book of life, but we're still trying to understand the grammar and syntax that makes those letters meaningful. We can fix obvious typos, mutations that clearly cause disease, but we're less certain about more subtle edits that might have far-reaching consequences. The story of CRISPR is ultimately a story about learning to read and write in the language of life itself. It's about discovering that the same bacterial immune system that protects microorganisms from viral infection can be repurposed to edit any genome with unprecedented precision. It's about realizing that the genetic code isn't fixed and immutable, but plastic and editable, at least if we're careful and wise about how we proceed. But it's also a story about humility. The more we learn about genetics, The more we realize how much we don't know, every answered question reveals 10 new mysteries. Every successful treatment highlights a dozen ways the intervention could go wrong. Every ethical dilemma we resolve uncovers 2 more that we hadn't considered. This isn't a reason for despair, it's a reason for wonder. We're living through one of the most extraordinary periods in human history, a time when we're gaining the power to consciously direct our own evolution. The decisions we make in the coming decades about how to develop and deploy CRISPR technology will echo through generations. Our children and grandchildren will inherit not just the genetic modifications we make, but the precedents we set for how such power should be wielded. In laboratories around the world tonight, as you settle into sleep, researchers are working with CRISPR systems that can edit genes with increasing precision and safety. They're developing new versions that can make temporary modifications rather than permanent ones. They're creating tools that can edit multiple genes simultaneously, or that can be activated only under specific conditions. They're even exploring whether CRISPR could be used not just to edit DNA, but to modify the epigenetic marks that determine which genes are turned on or off in different cells. Each of these advances brings new possibilities and new responsibilities. The ability to make temporary genetic modifications could allow for treatments that are reversible if problems arise. The capacity to edit multiple genes at once could make it possible to address complex genetic conditions that involve many different mutations. The option to control when and where genetic modifications are active could make treatments much safer and more precise. But with each new capability comes new questions about how it should be used and who should have access to it. The pace of scientific progress in CRISPR research shows no signs of slowing down, but our social and ethical frameworks for governing this technology are still catching up. We're in a race between technological capability and wisdom, between what we can do and what we should do. Perhaps that's why stories like Victoria Gray's are so important. They remind us that behind all the technical complexity and ethical debates are real people whose lives are transformed by this technology. Children who can finally leave the hospital, parents who no longer have to watch their loved ones suffer, families who can plan for the future without the shadow of genetic disease hanging over them. These personal stories don't resolve the larger questions about CRISPR, but they ground them in human experience. They remind us why we're developing these tools in the first place, not to play God or to pursue scientific knowledge for its own sake, but to reduce suffering and expand human flourishing. Every ethical framework we develop, every regulatory system we create, every international agreement we negotiate should keep these human stories at its center. The fascinating thing about CRISPR is how it emerged from the most unexpected place, not from grand laboratories funded by pharmaceutical giants, but from the humble world of yogurt production. Yes, you heard that right. Some of the most important early research on CRISPR was conducted by scientists working for Danisco, a company that makes cultures for yogurt and cheese. They were trying to solve a very practical problem: how to protect their bacterial cultures from viruses that could ruin entire batches of dairy products. It's rather poetic, isn't it? The technology that might one day cure genetic diseases was discovered by researchers trying to make better yogurt. It reminds us that scientific breakthroughs often come from the most mundane questions asked by curious minds who refuse to accept that's just how things work as a final answer. Dr. Rodolphe Barangu was one of those curious minds. Working in a small laboratory in France, he spent his days studying the bacterial cultures that transform milk into the creamy yogurt you might have enjoyed for breakfast. When viruses attacked these cultures, entire production runs would be lost, costing the company thousands of dollars. Barangu noticed that some bacterial strains seemed naturally resistant to certain viruses, and he wanted to understand why. What he discovered was that these resistant bacteria had incorporated pieces of viral DNA into their own genomes right between those mysterious CRISPR sequences that scientists had been puzzling over for decades. It was like finding that the bacteria were keeping a genetic mugshot file of their enemies, updating it every time they encountered a new threat. This discovery might have remained a footnote in the annals of dairy science, but Barangu and his colleagues realized they were looking at something much bigger. If bacteria could be programmed to remember and defend against specific viral sequences, could the same system be used to target any DNA sequence? Could it be turned into a general-purpose gene editing tool? The leap from dairy science to human therapeutics took several more years, and the contributions of researchers around the world. Jennifer Doudna at UC Berkeley and Emmanuel Charpentier, then at Umeå University in Sweden, figured out how to reprogram the CRISPR system to cut any desired DNA sequence. Feng Zhang at the Broad Institute showed that it could work in human cells. George Church at Harvard demonstrated its potential for treating genetic diseases. Each of these scientists built on the work of others, adding pieces to a puzzle that had been scattered across decades of research. It's a beautiful example of how science actually works, not as a series of lonely eureka moments, but as a collaborative conversation across time and space, with each researcher adding their voice to a growing chorus of understanding. By 2013, the scientific community was buzzing with excitement about CRISPR's potential. Papers were being published at a breathtaking pace, each one demonstrating new applications or improving the precision of the system. Venture capitalists began pouring money into CRISPR startups. Major pharmaceutical companies launched research programs. Universities filed patent applications and fought legal battles over who had invented what and when. But amid all the excitement and commercial interest, something important was happening in the quieter corners of the scientific world. Researchers were beginning to grapple with the profound ethical questions that CRISPR raised. If we could edit human genes, should we? If we could enhance human capabilities, who should decide what enhancements are acceptable? If we could make changes that would be passed down to future generations, how could we ensure we weren't making irreversible mistakes? In 2015, scientists from around the world gathered in Washington, D.C. for a summit on human gene editing. The meeting was unprecedented. Rarely had the scientific community paused to consider the ethical implications of a technology while it was still being developed, rather than after it had already been deployed. The discussions were intense, sometimes heated, but ultimately productive. The consensus that emerged was nuanced. Most participants agreed that using CRISPR to treat serious genetic diseases in individual patients, what scientists call somatic cell editing, was ethically justifiable and should be pursued with appropriate safeguards. But there was much less agreement about heritable editing changes that would be passed down to future generations. Some argued that heritable editing could prevent enormous suffering by eliminating genetic diseases from family lines entirely. Others worried that we didn't understand genetics well enough to make permanent changes to the human gene pool. Still, others questioned whether it was ethical to make irreversible modifications to people who couldn't consent to them, future generations who hadn't yet been born. The summit ended with a call for more research, more public engagement, and more international coordination. Participants agreed that heritable gene editing should not be attempted until there was broader scientific consensus about its safety and societal acceptance of its use. It was a thoughtful, measured response to an unprecedented situation. But science, as we know, doesn't always proceed at the measured pace that ethicists and policymakers might prefer. Technologies develop according to their own logic, driven by the curiosity of researchers, the needs of patients, and the dynamics of scientific competition. And sometimes, individual scientists decide to move faster than the scientific community as a whole thinks is wise. This brings us back to He Jiankui and the gene-edited twins that sent shockwaves through the scientific world in 2018. His actions were controversial, not just because he crossed ethical lines that the international community had agreed not to cross, but because of how he went about it. He worked in secret, didn't submit his research for peer review, and announced his results at a scientific conference rather than in a published paper where other researchers could scrutinize his methods. The scientific community's response was swift and decisive. Major journals refused to publish his work. Scientific societies condemned his actions. The Chinese government launched an investigation and eventually sentenced he to prison. International organizations called for stronger oversight of gene editing research. It was a dramatic reminder that science is not just about technical capability, but about community norms, ethical standards, and social responsibility. Yet the He Jiankui case also highlighted something important. The technical barriers to human gene editing are lower than many people realized. The equipment needed for CRISPR experiments costs thousands, not millions of dollars. The techniques can be learned by graduate students, not just senior researchers. The knowledge needed to attempt human gene editing is widely available in scientific journals and textbooks. This democratization of genetic engineering technology is both exciting and concerning. On one hand, it means that brilliant researchers in developing countries, in small universities, in startup companies, can contribute to solving humanity's greatest challenges. On the other hand, it means that the power to alter the human genome is spreading beyond the careful oversight of major research institutions. The question isn't whether we can prevent this democratization that ship has already sailed. The question is how we can foster a culture of responsibility among the growing community of CRISPR practitioners. How do we ensure that enthusiasm for genetic engineering is balanced with respect for its power? How do we encourage innovation while preventing harm? Some of the most promising answers are emerging from unexpected places. In laboratories across Africa, researchers are using CRISPR to develop crops that can survive drought and resist pests, potentially helping to feed millions of people as climate change makes agriculture more challenging. In clinics across Latin America, doctors are preparing to test CRISPR treatments for genetic diseases that disproportionately affect their populations. These applications share something important. They're focused on solving local problems using local expertise. Rather than waiting for solutions to be developed in wealthy countries and then exported around the world, communities are using CRISPR to address their own unique challenges. This represents a fundamental shift in how genetic engineering research is conducted and who gets to participate in it. But perhaps the most remarkable applications of CRISPR are happening not in human medicine, but in conservation biology. Scientists are exploring whether gene editing could help save species from extinction or help existing species adapt to rapidly changing environments. Could we edit the genes of coral to help them survive in warming oceans? Could we modify the immune systems of endangered animals to protect them from diseases introduced by human activities? These questions push the boundaries of our thinking about genetic modification even further. If we accept that it's ethical to edit human genes to prevent disease, is it also ethical to edit the genes of wild animals for their own protection? If we can help species adapt to climate change through genetic modification, do we have an obligation to do so? Or would such interventions represent an unacceptable level of human interference with natural processes? Dr. George Church at Harvard has proposed one of the most ambitious conservation applications of CRISPR, bringing back extinct species. His team is working on editing the genome of Asian elephants to incorporate genes from woolly mammoths with the goal of creating cold-adapted elephants that could help restore Arctic ecosystems. It's a project that sounds like science fiction, but is grounded in serious ecological science. The idea is that these mammoth-like elephants could help combat climate change by trampling down snow in the Arctic, exposing the frozen ground underneath to cold air, and helping to preserve the permafrost that stores enormous amounts of carbon. If the permafrost melts, that carbon gets released into the atmosphere, accelerating global warming. But if herds of cold-adapted elephants could help keep the permafrost frozen, they might play a role in slowing climate change. It's an audacious proposal that illustrates both the potential and the complexity of using CRISPR for conservation. On one hand, it represents a creative solution to a pressing environmental problem. On the other hand, it raises questions about whether we should be creating new hybrid species what happens if these creatures don't behave as we expect, and whether technological fixes can substitute for addressing the root causes of environmental problems. Similar questions arise with other conservation applications. Scientists are developing gene drives, genetic modifications that spread through wild populations on their own to combat malaria-carrying mosquitoes. The idea is to release genetically modified mosquitoes that either can't carry the malaria parasite or that produce offspring unable to survive. As these modified mosquitoes mate with wild populations, the genetic modifications spread, potentially eliminating malaria from entire regions. Field trials of gene-drive mosquitoes are already underway in several countries, with promising results. In some test areas, populations of malaria-carrying mosquitoes have declined dramatically. But the technology also raises concerns about unintended consequences. What happens if the genetic modifications spread beyond the target species? What if mosquito populations crash so dramatically that they affect other parts of the ecosystem? What if the modifications interact with other genetic variants in unexpected ways? These aren't theoretical concerns, they're urgent practical questions that researchers are grappling with as they develop and deploy gene-drive technologies. The stakes are enormous. Malaria kills over 400,000 people each year, mostly children in sub-Saharan Africa. Any technology that could eliminate or significantly reduce malaria transmission could save hundreds of thousands of lives. But the same technology could have far-reaching ecological consequences that are difficult to predict or control. The challenge is that ecosystems are incredibly complex, with intricate webs of relationships between species that we're still trying to understand. Removing or dramatically reducing one species, even one that causes human suffering, could have cascading effects throughout the ecosystem. Mosquitoes, for all the disease they carry, also serve as pollinators for some plants and as food for many animals. Birds, bats, fish, and other insects all depend on mosquitoes as part of their diet. This doesn't mean we shouldn't pursue gene-drive technologies for malaria control, but it does suggest that we need to proceed carefully with extensive testing and monitoring to detect any unintended consequences. It also highlights the importance of engaging local communities in decisions about whether and how to deploy these technologies. After all, the people who live in malaria-endemic areas are the ones who would benefit most from successful malaria elimination, but they would also bear the consequences if something went wrong. The same principles apply to other conservation applications of CRISPR. Australian researchers are exploring whether gene editing could help native species like the northern quoll develop resistance to the poisonous cane toads that are decimating their populations. American scientists are investigating whether CRISPR could help bats survive white nose syndrome, a fungal disease that has killed millions of bats across North America. Each of these projects requires careful consideration of the ecological, ethical, and social implications. Success in one context doesn't guarantee success in another. The genetic modification that saves one species might harm another. The intervention that works in a laboratory might fail in the wild, where conditions are far more complex and unpredictable. But perhaps the most profound questions raised by conservation applications of CRISPR are philosophical ones. If we can prevent extinctions through genetic modification, do we have an obligation to do so? If human activities are driving species to extinction, should we use genetic engineering to help them survive? Or should we focus on addressing the underlying causes of biodiversity loss? These questions don't have easy answers, and different cultures, different ethical traditions, and different scientific communities may reach different conclusions. Indigenous communities, who have often served as the most effective guardians of biodiversity, may have very different perspectives on genetic modification than urban scientists. Conservation biologists who have spent their careers studying natural ecosystems may view genetic interventions differently than molecular biologists who see them as elegant solutions to pressing problems. What's clear is that these decisions shouldn't be made by scientists alone, any more than decisions about human gene editing should be made only by doctors and researchers. The power to modify the genetic basis of life, whether human, animal, plant, or microbial, is too important to be left to any single group, no matter how well-intentioned or technically expert. This brings us to one of the most important challenges of our time. How do we make collective decisions about technologies that could reshape the biological world? How do we balance the potential benefits of genetic modification against the risks of unintended consequences? How do we ensure that the voices of all stakeholders, not just scientists and policymakers, but patients, communities, and future generations, are heard in these discussions? Some countries are experimenting with new forms of democratic participation in science, policy, bringing together diverse groups of citizens to learn about complex technologies and deliberate about how they should be regulated and deployed. These citizen panels, or consensus conferences, don't make binding decisions, but they provide valuable input to policymakers and help ensure that scientific development is responsive to broader social values. Other approaches focus on building public understanding of science, recognizing that informed citizens are better equipped to participate in democratic decisions about technology. Science museums, science cafes, and online platforms are creating new opportunities for people to learn about CRISPR and other genetic technologies in accessible, non-technical ways. Still, other initiatives focus on bringing together stakeholders from different backgrounds: scientists, ethicists, patients, community representatives, policymakers, to explore the implications of genetic technologies and develop shared frameworks for thinking about them. These multidisciplinary conversations are often messy and sometimes contentious, but they're essential for navigating the complex landscape of genetic modification. The goal isn't to reach unanimous agreement. That's probably impossible on issues as complex as genetic engineering. Rather, the goal is to ensure that decisions are made through inclusive, transparent processes that consider multiple perspectives and values. It's to create space for ongoing dialogue and course correction as our understanding of these technologies evolves. As you lie there in the quiet darkness, millions of these conversations are happening around the world. In research laboratories, scientists are debating the ethical implications of their experiments. In regulatory agencies, policymakers are wrestling with how to oversee technologies that are advancing faster than traditional regulatory frameworks can accommodate. In community centers and online forums, ordinary citizens are learning about genetic modification and discussing what it means for their families and communities. All of these conversations are part of the larger story of CRISPR, a story that includes not just the technical breakthroughs and medical applications, but also the social and ethical challenges of wielding such powerful technology responsibly. It's a story that's still being written, and one in which each of us has a role to play. Because ultimately, CRISPR isn't just a tool for scientists and doctors. It's a technology that will shape the biological world that we and our children inherit. The decisions we make about how to develop and deploy it will echo through generations. Our choices about which genetic modifications to pursue and which to avoid, about who gets access to genetic therapies and who doesn't, about how to balance innovation with precaution, will help determine what kind of future we create. This isn't to say that every person needs to become a genetic engineer or a bioethicist, but it is to say that we all have a stake in how these technologies develop, and we all have a voice in the conversations that will shape their future. Whether through voting, community engagement, charitable giving, or simply staying informed about scientific developments, each of us can contribute to ensuring that the power to edit the book of life is used wisely and well. The story of CRISPR is in many ways a story about human potential, our capacity for curiosity, creativity, and collaboration in the face of enormous challenges. It's a story about how knowledge accumulated over decades of basic research can suddenly crystallize into transformative applications. It's about how technologies developed in one context can find unexpected uses in completely different fields. But it's also a story about human responsibility, our obligation to consider not just what we can do, but what we should do. It's about the importance of proceeding thoughtfully and inclusively when we're dealing with technologies that could have far-reaching consequences. It's about recognizing that scientific power comes with social obligations. As CRISPR continues to evolve, we can expect to see applications that we can barely imagine today. Scientists are already exploring whether the technology could be used to enhance human longevity, to improve cognitive function, or to help our species adapt to the challenges of space exploration. They're investigating whether CRISPR could help us develop new materials, new energy sources, or new ways of cleaning up environmental contamination. Some of these applications will succeed spectacularly, solving problems that have plagued humanity for generations. Others will fail or prove to have unacceptable side effects. Still, others will succeed technically, but prove to be socially or ethically problematic. The challenge is distinguishing between these different possibilities before we've invested enormous resources or created irreversible changes. This is why the scientific community's emphasis on careful, step-by-step development of CRISPR technologies is so important. Every successful clinical trial, every safety study, every ethical review brings us closer to understanding both the potential and the limitations of genetic modification. Every failed experiment teaches us something valuable about how these systems work and how they might be improved. The pace may sometimes seem frustratingly slow, especially for patients and families affected by genetic diseases. When your child is suffering from a condition that could theoretically be cured with CRISPR, waiting for more research and more safety data can feel like an eternity. But the careful, methodical approach that characterizes the best scientific research is designed to maximize the chances that genetic therapies will be both safe and effective when they finally reach patients. This is particularly important because genetic modifications, once made, can be difficult or impossible to reverse. Unlike traditional drugs, which are gradually metabolized and eliminated from the body, genetic changes can persist for months, years, or even lifetimes. A mistake in genetic therapy could have consequences that last far longer than a mistake in conventional treatment. But when genetic therapies work, they can be transformative in ways that conventional treatments cannot match. Instead of managing symptoms or slowing the progression of disease, they can address the root genetic causes of illness. Instead of requiring daily medications or frequent medical procedures, they can provide cures that last for years or decades with a single treatment. This is what makes the development of CRISPR therapies so exciting and so challenging. The potential benefits are enormous, but so are the risks of getting it wrong. The scientific community is working hard to maximize the former while minimizing the latter, but it's a delicate balance that requires constant attention and adjustment. As you settle deeper into the gentle embrace of sleep, consider this remarkable truth. Every person on Earth carries within them a story written in DNA that stretches back billions of years. Your genetic code contains echoes of ancient bacteria, traces of our earliest ancestors, and the accumulated wisdom of countless generations who survived long enough to pass their genes forward. It's a library of survival, written in 4 simple letters, housed in every cell of your being. And now, for the first time in all of human history, we're learning not just to read this ancient text, but to edit it with care and precision. CRISPR represents a profound shift in our relationship with our own biology, from passive recipients of genetic inheritance to active curators of our genetic future. But this power comes with questions that reach far beyond the laboratory. In fertility clinics around the world, couples are beginning to ask whether they should screen their embryos for genetic diseases or even enhance their children's genetic potential. The technology exists to test embryos for hundreds of genetic conditions before implantation. Soon, it may be possible to correct genetic problems directly, ensuring that children are born free from inherited diseases that have plagued their families for generations. This pre-implantation genetic diagnosis, or PGD, is already routine in many fertility clinics. Couples who know they carry genes for serious inherited diseases can have their embryos tested, selecting only those free from genetic problems for implantation. It's a form of genetic selection that's already changing the landscape of human reproduction, even without direct gene editing. But CRISPR takes this possibility much further. Instead of simply selecting embryos that happen to be free from genetic problems, we could potentially correct those problems directly, turning affected embryos into healthy ones. We could even go beyond correction to enhancement, adding genetic variants that provide resistance to disease, improved cognitive function, or other desirable traits. The technical challenges of embryo editing are significant but not insurmountable. Human embryos are delicate, and the timing of genetic modifications is critical. The changes must be made early enough to affect all cells of the developing organism, but not so early that they interfere with normal development. The CRISPR system must work with extraordinary precision, avoiding off-target effects that could cause new problems while solving old ones. Recent advances in CRISPR technology have dramatically improved its accuracy and safety. New versions of the system can make single-letter changes in DNA with unprecedented precision. Scientists have developed methods for delivering CRISPR components to cells more effectively and for controlling when and where genetic modifications occur. The technical barriers to safe, effective human embryo editing are falling rapidly. But technical feasibility doesn't resolve the deeper questions about whether such interventions are wise or ethical. Embryo editing affects not just the individuals born from modified embryos, but all of their descendants. A genetic change made today could echo through hundreds of generations, affecting people who will live centuries from now. How can we possibly predict all the consequences of such interventions? The history of medicine is littered with treatments that seemed promising at first, but later proved harmful. The drug thalidomide was prescribed to pregnant women to prevent morning sickness, but it caused severe birth defects in thousands of children. The hormone DES was given to prevent miscarriages, but increased cancer risk in the daughters of women who took it. Even more recently, some gene therapy trials have resulted in serious side effects, including cancer caused by the very vectors used to deliver therapeutic genes. These examples highlight the importance of proceeding carefully with any medical intervention, but especially with genetic modifications that could have lifelong or even multi-generational effects. The scientific community has learned hard lessons about the need for rigorous safety testing, careful monitoring of long-term effects, and honest acknowledgement of uncertainty about new treatments. Yet the potential benefits of embryo editing are equally compelling. Imagine a world where no child is born with cystic fibrosis, where Huntington's disease is eliminated from families that have suffered from it for generations, where genetic forms of blindness and deafness become historical curiosities rather than current realities. The suffering prevented by safe, effective embryo editing could be immense. Some genetic diseases cause not just individual suffering but enormous social costs. A child born with severe intellectual disabilities may require lifelong care costing millions of dollars. A family dealing with a degenerative genetic disease may face not just emotional trauma but financial ruin. From a purely utilitarian perspective, the resources saved by preventing genetic diseases could be redirected toward other social goods, education, infrastructure, environmental protection. But utilitarian calculations, however compelling, don't capture the full complexity of these decisions. Each genetic variant tells a story about human diversity and the paths that evolution has taken. Some variants that cause disease in certain contexts provide benefits in others. The genetic changes that cause sickle-cell disease also provide resistance to malaria. The genes associated with autism may also contribute to exceptional abilities in pattern recognition and systematic thinking. This genetic complexity means that Decisions about which variants to eliminate and which to preserve aren't simply medical decisions, they're decisions about what kinds of human diversity we value and want to maintain. They reflect our beliefs about what constitutes a life worth living and what kinds of differences we should celebrate rather than cure. The deaf community has been particularly eloquent in articulating these concerns. Many deaf individuals don't experience their deafness as a disability but as a cultural identity. They have their own language, their own community, their own ways of experiencing and understanding the world. If genetic deafness were eliminated through embryo editing, would something valuable be lost? Would we be practicing a subtle form of cultural genocide? Similar questions arise around other conditions. The autism community includes many individuals who view their neurological differences not as disorders to be cured, but as alternative ways of thinking and experiencing the world. Some have made extraordinary contributions to science, art, and technology precisely because their brains work differently from the neurotypical majority. Even conditions that most people would clearly classify as diseases raise complex questions when viewed through the lens of genetic editing. Huntington's disease causes devastating neurodegeneration, but it typically doesn't manifest until midlife, after people have had decades to live, love, work, and contribute to society. Some individuals at risk for Huntington's choose not to be tested, preferring to live with uncertainty rather than the knowledge of their genetic fate. Should we make that choice for them through embryo editing? These dilemmas become even more complex when we move beyond treating disease to enhancing normal human capabilities. The genetic factors that influence intelligence, athletic ability, disease resistance, and longevity are increasingly well understood. In principle, CRISPR could be used not just to prevent genetic diseases but to enhance desirable traits. Some enhancements might seem relatively uncontroversial. Most parents would probably welcome genetic modifications that reduced their children's risk of heart disease, cancer, or dementia. But what about modifications that increased IQ by 10 points, or that provided resistance to HIV infection, or that extended healthy lifespan by decades? The line between treatment and enhancement isn't as clear as it might seem. Many of the genetic variants we think of as normal actually increase disease risk compared to other variants. Is correcting a genetic variant that increases heart disease risk by 20% a treatment or an enhancement? What about one that increases risk by 5%? Where exactly do we draw the line between fixing genetic problems and improving genetic potential? These questions become even more challenging when we consider that genetic enhancements, like genetic treatments, could exacerbate existing inequalities. If genetic modifications are expensive and not covered by insurance, they could become luxury goods available only to the wealthy. Over time, this could create genetic class divisions, with the genetically enhanced enjoying advantages in health, intelligence, and longevity that are literally written into their DNA. The prospect of genetic inequality is particularly troubling because, unlike other forms of inequality, it could be passed down through generations. A family that could afford genetic enhancements for their children would pass those advantages on to their grandchildren and great-grandchildren. Over time, genetic haves and have-nots could diverge into essentially different subspecies. Some philosophers and ethicists argue that we have a moral obligation to prevent such genetic stratification by ensuring that genetic enhancements, if they're developed, are made widely available. Others contend that genetic enhancement is inherently problematic because it treats children as projects to be optimized rather than individuals to be loved unconditionally. Still, others argue that the focus on genetic enhancement distracts from more pressing social problems like poverty, education, and healthcare access. These debates are happening not just in academic journals, but in fertility clinics, where couples are already making decisions about genetic testing and selection. As CRISPR technology advances, these decisions will become more complex and consequential, genetic counselors are beginning to grapple with how to help families navigate choices that previous generations never had to face. Dr. Sarah Chen, a genetic counselor at a major fertility clinic, spends her days helping couples understand their genetic risks and options. She's seen firsthand how CRISPR is changing the landscape of reproductive choice. 10 years ago, couples who carried genes for serious genetic diseases had limited options, she explains. They could take their chances with natural conception, use donor eggs or sperm, adopt, or remain childless. Now, we're beginning to offer them the possibility of having genetically related children who are free from the diseases they carry. But Dr. Chen also sees the challenges that come with expanded options. When there are more choices, decisions become harder, she notes. Couples feel pressure to make the right choice for their children, but there often isn't a clear right answer. Every option involves trade-offs, and we're asking people to make decisions about technologies they don't fully understand, with consequences we can't fully predict. The psychological burden of genetic choice is becoming an important area of research. Some couples report feeling empowered by having options, while others describe feeling overwhelmed by the responsibility of making genetic decisions for their children. Still, others worry about the message that genetic selection sends to existing family members who have genetic conditions. Consider a couple where one partner has achondroplasia, a form of dwarfism. They're planning to have children and learn that they could use CRISPR to ensure their children don't inherit the condition. But the partner with achondroplasia doesn't view their condition as something that needs to be fixed. They've lived a full, rich life, have a successful career, and are proud of their identity as a person with dwarfism. What message would genetic corrections send to them? That their life isn't worth living? That they're something their children should be protected from becoming? These personal dilemmas reflect broader social tensions about disability, diversity, and the goals of medicine. The medical model tends to view genetic conditions as problems to be solved, while the social model recognizes that many challenges faced by people with genetic differences stem from social barriers rather than inherent limitations. CRISPR technology has the potential to eliminate many genetic conditions, but it also raises questions about whether elimination is always the best approach. Some advocates argue for a middle path that focuses on improving treatments and social support for people with genetic conditions, while also developing genetic therapies for those who want them. Others contend that resources would be better spent on addressing environmental causes of disease and improving healthcare access for all. Still, others believe that genetic enhancement could help create a healthier, more capable human species better equipped to face future challenges. As you lie there in the quiet darkness, these debates about our genetic future are unfolding in laboratories, clinics, and policy forums around the world. The decisions being made today about CRISPR research and regulation will shape the options available to your children and grandchildren. They'll determine whether genetic modification becomes a routine part of healthcare or remains an experimental treatment available only to a few. The pace of scientific progress means that some of these decisions can't wait for perfect consensus. Clinical trials of CRISPR therapies are already underway, and their results will influence how quickly the technology moves from experimental treatment to standard care. Regulatory agencies are developing frameworks for overseeing genetic modifications, even as the science continues to evolve rapidly. International coordination is becoming increasingly important as CRISPR research advances. A treatment developed in one country could affect people around the world, especially if it involves heritable genetic modifications. Different countries have different ethical standards and regulatory approaches, creating the potential for genetic tourism where people travel to countries with more permissive policies. Some nations are positioning themselves as leaders in genetic medicine, investing heavily in CRISPR research, and creating regulatory frameworks designed to encourage innovation. Others are taking more cautious approaches, implementing strict oversight and lengthy approval processes. Still, others have banned certain types of genetic modification entirely, at least for now. These different national approaches reflect different cultural values, risk tolerances, and priorities. Countries with socialized healthcare systems may be more willing to invest in expensive genetic therapies if they could reduce long-term healthcare costs. Nations with strong religious traditions may be more skeptical of genetic modifications that seem to interfere with natural or divine processes. Developing countries may prioritize genetic modifications that address diseases prevalent in their populations. The global nature of genetic modification research means that these different approaches will inevitably interact and influence each other. Scientific knowledge crosses borders easily, but regulatory frameworks and ethical standards don't. A genetic modification developed in one country with permissive regulations could be used in countries with stricter policies, or vice versa. Patients might travel internationally to access treatments not available in their home countries. This international dimension adds another layer of complexity to already challenging decisions about genetic modification. It raises questions about justice and equity on a global scale. Should genetic enhancements be available to anyone who can afford them, regardless of their country's policies? Should countries with more restrictive approaches be able to prevent their citizens from accessing genetic modifications available elsewhere? Some international organizations are working to develop global standards for genetic modification research and applications. The World Health Organization has established an expert committee on human genome editing to develop recommendations for governance and oversight. Scientific organizations are collaborating to establish international registries of genetic modification research and to share safety data across borders. But achieving meaningful international coordination on genetic modification will require more than technical cooperation. It will require navigating deep differences in values, priorities, and governance systems. What seems like an obvious ethical principle in one cultural context may be controversial or even offensive in another. The challenge is finding common ground, while respecting legitimate differences in how societies approach these technologies. As CRISPR continues to advance, we can expect these international dimensions to become increasingly important. Genetic modifications don't respect national borders, and the benefits and risks of these technologies will be shared globally, whether we plan for it or not. Developing effective international governance of genetic modification may be one of the most important challenges of our time. Meanwhile, in laboratories around the world, scientists continue to push the boundaries of what's possible with CRISPR technology. They're developing new versions that are more precise, more efficient, and safer. They're exploring applications beyond genetic diseases to cancer, aging, and enhancement. They're investigating whether CRISPR could be used not just to edit DNA, but to modify the epigenetic marks that control gene expression. Each advance brings new possibilities and new questions. Prime editing, a newer version of CRISPR, can make precise insertions, deletions, and substitutions in DNA with minimal off-target effects. Base editing can change single letters of genetic code without creating double-strand breaks in DNA. These technical improvements are making genetic modifications safer and more precise, addressing some of the concerns about CRISPR therapy. But technical advances also raise new possibilities that challenge existing ethical frameworks. If genetic modifications become precise enough to be essentially risk-free, do the ethical calculations change? If the costs of genetic therapies drop dramatically, does that alter considerations about access and equity? If we develop genetic modifications that can be turned on and off at will, does that address concerns about irreversibility? Scientists are also exploring applications of CRISPR that go far beyond editing genes in individual patients. They're investigating whether CRISPR could be used to create universal donor organs that wouldn't trigger immune rejection. They're developing genetic modifications that could help human beings adapt to the challenges of space exploration, resistance to radiation, ability to survive in low oxygen environments, enhanced bone density to prevent problems in zero gravity. Some researchers are even exploring whether CRISPR could be used to address aging itself. By editing genes associated with longevity, or by modifying cellular repair mechanisms, it might be possible to extend healthy human lifespan significantly. Laboratory studies have already shown that genetic modifications can dramatically extend lifespan in mice and other animals. Whether similar approaches could work in humans remains to be seen. The prospect of using genetic modification to extend human lifespan raises profound questions about the nature of human existence. If people could live for 150 years, or 200 years, or even longer, how would that change society? How would it affect careers, relationships, family structures? Would it exacerbate inequality by giving the genetically enhanced not just better health, but dramatically longer lives to accumulate wealth and power? These questions may seem like science fiction, but they're beginning to influence research priorities and policy discussions today. Some countries are investing heavily in longevity research, viewing it as a key to maintaining economic competitiveness as their populations age. Others are more skeptical, questioning whether dramatically extended lifespans would be beneficial for individuals or society. The intersection of CRISPR technology with artificial intelligence is opening up even more possibilities. AI systems can analyze vast genetic datasets to identify potential targets for genetic modification. They can predict the effects of genetic changes with increasing accuracy. They can even help design new CRISPR systems with improved precision and efficiency. Machine learning algorithms are already being used to optimize CRISPR guide RNAs, reducing off-target effects and improving editing efficiency. AI systems can analyze the genetic variations present in different populations to design personalized genetic therapies. They can even help predict which patients are most likely to benefit from genetic modifications. But the combination of genetic modification and artificial intelligence also raises new concerns about privacy, autonomy, and control. If AI systems can predict genetic diseases before they manifest, or identify genetic variants associated with behavioral traits, how should that information be used? Who should have access to genetic predictions? And how should they be regulated? The integration of genetic and digital technologies is creating new forms of biological surveillance that were unimaginable just a few years ago. Genetic information is increasingly being combined with other health data, behavioral data, and even social media activity to create comprehensive profiles of individuals' health risks and likely outcomes. This information could be used to personalize medical treatment, but it could also be used for discrimination, manipulation, or social control. These technological convergences are happening faster than our regulatory and ethical frameworks can adapt. We're creating capabilities that no previous generation has possessed, but we're doing so with governance systems designed for earlier, simpler technologies. The challenge is developing new approaches to oversight and accountability that can keep pace with rapid technological change while preserving the values we care about. As you drift closer to sleep, consider that you're living through one of the most extraordinary periods in human history. We're gaining the power to consciously direct our own biological evolution, to edit the genetic instructions that have guided life for billions of years. The decisions we make about how to use this power will echo through generations, shaping the biological future of our species and our planet. But you don't need to solve all these complex questions tonight. The scientific community, policymakers, ethicists, and citizens around the world are working to ensure that CRISPR technology is developed and deployed responsibly. Your role is to stay informed, to engage in these discussions when you can, and to support approaches that reflect your values and priorities. The story of CRISPR is still being written, and it's a story in which we all have a part to play. Whether through the medical choices we make for ourselves and our families, the policies we support, or simply the conversations we have about these technologies, each of us contributes to shaping how genetic modification evolves. In the gentle darkness of your room, as your cells continue their ancient work of maintenance and repair, millions of researchers around the world are laboring to understand and improve the molecular machinery of life. They're driven by the same curiosity that led to the discovery of CRISPR in bacterial cultures, the same desire to reduce suffering and expand human possibility that motivates all of medicine. The bacterial immune system that inspired CRISPR has been operating for billions of years, a testament to evolution's capacity for elegant solutions to complex problems. Now we're learning to harness that ancient wisdom, to use the tools that life has developed to edit life itself. It's a profound responsibility and an extraordinary opportunity, the chance to become conscious participants in the ongoing story of evolution. Tomorrow morning, when you wake and stretch and feel your muscles respond to your brain's gentle commands, billions of genetic instructions will already have been at work for hours. Your liver will have processed the remnants of yesterday's meals using enzymes coded by genes that have remained largely unchanged for millions of years. Your immune cells will have patrolled your bloodstream, each one carrying a molecular memory of every pathogen you've ever encountered, thanks to genetic programs more sophisticated than any computer virus scanner we've ever designed. But scattered throughout those same cells, in laboratories around the world, scientists are conducting experiments that would have seemed like pure magic to researchers just 20 years ago. They're taking cells from patients with genetic diseases, editing out the mutations that cause suffering, and returning those cells to heal bodies that were once thought incurable. They're engineering bacteria to produce life-saving medicines, plants to survive droughts that would have destroyed entire harvests, and even mosquitoes that could eliminate diseases that have plagued humanity for millennia. The elegance of it is almost overwhelming when you really stop to think about it. CRISPR doesn't force its way into cells like some molecular battering ram. Instead, it slips in quietly, guided by RNA sequences that act like cellular GPS coordinates, leading it precisely to the genetic address it needs to visit. Once there, it makes its edit with the careful precision of a master librarian, correcting a single typo in a vast collection, leaving everything else exactly as it found it. Dr. Feng Zhang, one of the pioneers who helped bring CRISPR into human cells, likes to describe the feeling of watching the system work for the first time. It was like seeing a lock click open that we'd been trying to pick for decades, he says. Suddenly, we could go anywhere in the genome, make any change we wanted, with a precision that seemed almost too good to be true. But perhaps what's most remarkable about CRISPR isn't its precision, it's its accessibility. Unlike previous genetic engineering tools that required massive laboratories, teams of specialists, and budgets that only pharmaceutical giants could afford, CRISPR can be learned by undergraduate students and performed with equipment that fits on a laboratory bench. This democratization of genetic engineering is quietly revolutionizing who gets to participate in editing the Book of Life. In a small laboratory in Nairobi, researchers are using CRISPR to develop crops that can survive the increasingly unpredictable rainfall patterns brought by climate change. In Sao Paulo, scientists are engineering mosquitoes that could eliminate dengue fever from urban areas. In rural Bangladesh, plant breeders are creating rice varieties that can grow in increasingly salty soils as sea levels rise and saltwater intrudes into agricultural areas. Each of these applications shares something profound. They're solutions developed by people who understand local problems intimately, using tools that were once available only to researchers in wealthy countries. It represents a fundamental shift in how genetic engineering research is conducted from a centralized enterprise controlled by major institutions to a distributed effort involving scientists and communities around the world. But this democratization also brings new responsibilities. When genetic engineering was confined to a few major laboratories with extensive oversight and safety protocols, the risks were relatively contained. Now, as CRISPR spreads to smaller labs, community spaces, and even home workshops, ensuring that experiments are conducted safely and ethically becomes much more challenging. The question isn't whether we can prevent this spread of capability that wave has already crested and is flowing around the world. The question is how we foster a culture of responsibility among the growing community of genetic engineers. How do we ensure that enthusiasm for solving problems with CRISPR is balanced with respect for its power? How do we encourage innovation while preventing harm? Some of the most thoughtful answers are emerging from unexpected places. In community laboratories across cities like Boston, London, and Tokyo, citizen scientists are developing their own codes of conduct for genetic engineering experiments. They're creating peer review systems, safety protocols, and educational programs that help ensure their work meets high standards of scientific rigor and ethical responsibility. These grassroots efforts reflect something important about how transformative technologies actually spread through society. It's rarely a top-down process driven by major institutions and government policies. Instead, it's usually a bottom-up phenomenon driven by countless individuals who find creative ways to use new tools to solve problems they care about. The internet revolution happened not because telecommunications companies decided it should, but because millions of people found ways to connect, share information, and build communities online. Similarly, the genetic engineering revolution is being driven not just by pharmaceutical companies and university researchers, but by farmers trying to feed their communities, doctors treating rare diseases, and conservationists trying to save endangered species. This distributed innovation brings both tremendous opportunities and significant risks. On the opportunity side, it means that brilliant minds everywhere can contribute to solving humanity's greatest challenges. The next breakthrough in cancer treatment might come not from a major research university, but from a small laboratory in Lagos or Lima. The genetic modification that helps crops survive climate change might be developed by farmers in rural India or Kenya. On the risk side, it means that genetic engineering is being conducted by people with varying levels of training, resources, and oversight. A mistake in a major pharmaceutical laboratory might affect the patients enrolled in a clinical trial. A mistake in a community laboratory or home workshop could have consequences that are much harder to predict or contain. This is why education and community engagement are becoming so crucial to the responsible development of CRISPR technology. People need to understand not just how these tools work, but why they matter and how they might affect their communities. They need opportunities to participate in discussions about how genetic engineering should be regulated and deployed, and they need ways to hold researchers and institutions accountable for the safety and ethics of their work. Science museums around the world are creating new exhibits that let visitors explore CRISPR through interactive displays and hands-on activities. Online platforms are offering courses that teach the basics of genetic engineering to anyone with an internet connection. Community forums are bringing together scientists, ethicists, patients, and ordinary citizens to discuss the implications of genetic modification. But perhaps most importantly, these educational efforts are helping people understand that genetic engineering isn't something being done to them by distant experts. It's something they can participate in, influence, and shape according to their values and priorities. The future of CRISPR won't be determined solely by scientists and policymakers. It will be shaped by the collective choices of communities around the world. Consider the story of Josiah Zayner, a former NASA scientist who became one of the most visible advocates for DIY genetic engineering. In 2017, he made headlines by injecting himself with CRISPR, designed to enhance muscle growth, a dramatic demonstration of how accessible the technology had become. While many scientists criticized his stunt as dangerous and irresponsible, it sparked important conversations about who gets to decide how genetic engineering should be used. Zayner's company, the Odin, sells CRISPR kits that let people conduct genetic engineering experiments at home. The kits come with bacteria, genetic templates, and all the equipment needed to modify genes in a kitchen or garage. They're designed to be educational rather than therapeutic. You can make bacteria glow green or produce human insulin, but you can't cure genetic diseases with them. Still, the very existence of such kits illustrates how far genetic engineering has come from its origins in highly controlled laboratory settings. Today, a curious teenager with a few $100s can perform experiments that would have required a PhD and $1 million laboratory just 20 years ago. This accessibility is both thrilling and concerning. On one hand, it's democratizing one of the most powerful technologies ever developed, making it available to people who might never have had the opportunity to participate in genetic engineering research. On the other hand, it's putting tools with enormous potential for both good and harm into the hands of people who may not fully understand their implications. The challenge is finding ways to preserve the benefits of open, distributed innovation, while minimizing the risks of accidents or misuse. This requires not just better safety protocols and regulatory oversight, but also cultural changes in how we think about scientific research and technological development. Traditional scientific culture emphasizes peer review, institutional oversight, and careful publication of results. DIY culture emphasizes experimentation, iteration, and open sharing of methods and findings. These two approaches can complement each other, but they can also clash in ways that create confusion and conflict. Some established scientists worry that DIY genetic engineering undermines the careful safety and ethical standards that the scientific community has developed over decades. Some DIY practitioners feel that institutional science is too slow, too conservative, and too removed from the real-world problems they're trying to solve. Bridging these cultural divides will require patience, understanding, and a willingness to learn from each other. Established scientists need to recognize that distributed innovation can accelerate progress in ways that centralized research cannot. DIY practitioners need to appreciate the value of the safety protocols and ethical frameworks that the scientific community has developed through hard-won experience. Perhaps most importantly, both communities need to work together to ensure that genetic engineering develops in ways that benefit everyone, not just those with access to the latest technologies or the resources to develop them. This means thinking carefully about issues of equity, access, and justice as CRISPR applications move from experimental treatments to routine medical care. The cost of early CRISPR therapies illustrates this challenge starkly. Current treatments can cost hundreds of thousands of dollars per patient, putting them out of reach for most people around the world. If genetic medicine develops primarily as a luxury good available only to the wealthy, it could create new forms of inequality that are literally written into our DNA. Some countries are addressing this challenge by investing heavily in making genetic therapies available through public health systems. The United Kingdom's National Health Service has announced plans to provide CRISPR treatments for certain genetic diseases at no cost to patients. Other countries are exploring innovative financing mechanisms that could make expensive genetic therapies more affordable. But ultimately, achieving equitable access to genetic medicine will require not just creative financing, but also technological innovations that drive down costs. This is where the democratization of genetic engineering could have its most important impact. As more people around the world gain the ability to develop and produce CRISPR treatments, competition could drive prices down and make genetic medicine more widely available. Open source approaches to genetic engineering could play a particularly important role in this process. Just as open source software has made computing more accessible and affordable around the world, open source approaches to genetic engineering could help ensure that the benefits of CRISPR are widely shared. Several organizations are already working to develop open source genetic engineering tools and protocols. The BioBricks Foundation maintains a registry of standardized biological parts that anyone can use to build genetic systems. The SBOL, Synthetic Biology Open Language, initiative is developing standardized ways to describe genetic modifications that can be shared across different laboratories and platforms. These efforts reflect a growing recognition that genetic engineering is too important to be controlled by any single organization or country. The challenges that CRISPR could help address genetic diseases, climate change, food security are global in scope and require global solutions. Developing these solutions will require unprecedented levels of collaboration and knowledge sharing. As you lie there in the gentle darkness, your body performing its nightly maintenance routines with the precision of billions of years of evolutionary refinement, Researchers around the world are working to understand and improve upon nature's elegant solutions to the challenges of life. They're driven by the same curiosity that led to penicillin, vaccines, and countless other medical breakthroughs that have extended and improved human lives. But CRISPR represents something qualitatively different from previous medical advances. Instead of fighting diseases or managing symptoms, it offers the possibility of preventing genetic conditions entirely by correcting them at their source. Instead of treating patients one at a time, it could potentially eliminate inherited diseases from family lines forever. This shift from treatment to prevention, from individual therapy to population-level intervention, raises profound questions about the goals of medicine and the nature of human flourishing. If we can prevent genetic diseases, should we focus our efforts on doing so, or should we continue to invest in treatments and support systems for people who have genetic conditions? The disability rights community has been particularly thoughtful about these questions. Many people with genetic conditions don't view themselves as broken or in need of fixing. They've built rich communities, developed unique perspectives, and made valuable contributions to society precisely because their experiences have been different from the mainstream. Amanda Leduc, a writer with Spina Bifida, captures this complexity beautifully. "I don't want to be cured," she says. "I want to be accommodated. I want the world to be designed in ways that include people like me, not in ways that assume we shouldn't exist." Her perspective highlights one of the most challenging aspects of genetic prevention: the tension between reducing suffering and preserving diversity. Most people would agree that preventing severe, painful genetic conditions is a worthy goal. But what about conditions that cause challenges without necessarily causing suffering? What about genetic variants that create differences without necessarily creating disabilities? These questions don't have easy answers, and they're questions that each family, each community, and each society will need to grapple with as genetic prevention becomes more widely available. The decisions we make will reflect our deepest values about what kinds of differences we celebrate and what kinds of challenges we're willing to help people navigate. But perhaps the most important thing to remember is that these decisions don't have to be made in isolation or by experts alone. CRISPR technology is developing in a democratic context where patients, families, communities, and citizens can influence how it's used. The future of genetic medicine won't be determined solely by what's technically possible, but by what we collectively decide is desirable and appropriate. This is both a tremendous opportunity and a significant responsibility. We have the chance to shape one of the most powerful technologies ever developed in ways that reflect our values and priorities. But we also have the obligation to engage thoughtfully with these choices, to consider their implications for future generations, and to ensure that the benefits of genetic medicine are shared equitably. The conversation about CRISPR's future is happening right now in laboratories, clinics, policy forums, and communities around the world. Scientists are conducting research that will determine which applications are safe and effective. Regulators are developing frameworks that will govern how genetic modifications can be developed and deployed. Ethicists are exploring the moral implications of editing human genes. Patients and families are making personal decisions about whether to participate in clinical trials or use genetic testing services. Each of these conversations is part of the larger story of how CRISPR will develop and how it will be integrated into human society. And each of us has a voice in that story, whether through the medical choices we make, the policies we support, the research we fund, or simply the conversations we have with friends, family, and neighbors about these technologies. The bacterial immune system that inspired CRISPR operated for billions of years without any conscious oversight or ethical review. It evolved through the simple logic of survival. Bacteria that could defend themselves against viruses were more likely to reproduce and pass on their genes. The system worked because it was tested continuously by evolutionary pressure and refined through countless generations of trial and error. We don't have the luxury of billions of years to perfect genetic engineering technologies through evolutionary trial and error. We're developing these tools on human timescales, making conscious decisions about how they should work and how they should be used. This requires us to be both ambitious and humble, ambitious enough to pursue the tremendous potential of genetic medicine, but humble enough to acknowledge the limits of our knowledge and the possibility of unintended consequences. The story of CRISPR is ultimately a story about learning to read and write in the language of life itself, about discovering that the genetic code isn't fixed and immutable but editable and improvable. It's about realizing that we're not just passive recipients of our genetic inheritance, but active participants in shaping the biological future of our species and our planet. As you settle deeper into the embrace of sleep, your cells continuing their ancient dance of maintenance and renewal, know that you're part of this extraordinary moment in human history. The genetic instructions that guide your body's functions tonight are the product of billions of years of evolution, tested and refined through countless generations of our ancestors, but they're also, for the first time, potentially editable by conscious choice rather than random mutation. This power brings both tremendous opportunities and significant responsibilities. The choices we make about genetic engineering in the coming decades will echo through generations, shaping the biological legacy we leave to our children and grandchildren. But we don't have to make these choices alone or in haste. We have communities of scientists, ethicists, patients, and citizens working to ensure that genetic medicine develops in ways that reflect our highest values and aspirations. The gentle hum of life continues all around you in every cell division, every protein synthesis, every immune response. And now, for the first time, we're learning to join that ancient conversation, to add our own careful edits to the book of life. It's a privilege and a responsibility, unlike any our species has ever faced, and it's unfolding quietly, methodically, with the same patient precision that has guided evolution for billions of years. In laboratories scattered across the globe, researchers are working late into the night, driven by the same curiosity that led ancient observers to wonder about the nature of life itself. They're asking new questions about old mysteries, developing new tools to address ancient challenges, and opening new chapters in the continuing story of what it means to be alive in an age when life itself has become editable. The story continues, and you're part of it, whether you know it or not. Every breath you take, every heartbeat, every thought that drifts through your mind as you approach sleep is part of the grand experiment of conscious life, engaging with its own biological foundations. And tomorrow, when you wake refreshed and renewed, that experiment will continue, one careful edit at a time. Rest well, dear listener, and dream gently of the remarkable journey we've taken together tonight. We've wandered through the discovery of CRISPR in humble bacterial cultures, marveled at its transformation into humanity's most precise gene-editing tool, and pondered the profound questions it raises about our genetic future. From the yogurt laboratories where scientists first noticed those curious repetitive sequences, to the cutting-edge clinics where patients like Victoria Gray are experiencing miraculous cures, we've seen how scientific curiosity can blossom into technologies that reshape the very essence of what it means to be human. We've explored the elegant simplicity of nature's own genetic scissors, and witnessed how researchers learn to reprogram this ancient bacterial defense system into a tool for healing. Along the way, we've grappled with questions that would have seemed like pure philosophy just decades ago, but now demand urgent, practical answers. Should we edit the genes of future generations? How do we balance the promise of genetic enhancement with concerns about equality and justice? What happens when the power to modify life itself becomes as accessible as a smartphone app? These aren't questions with easy answers, and perhaps that's as it should be. The most profound technologies often arrive accompanied by profound uncertainties. But what strikes me as deeply hopeful is how thoughtfully the scientific community has approached these challenges, pausing to consider ethics, even as technical capabilities race forward, engaging with diverse voices and perspectives, recognizing that the power to edit life belongs not to any single group, but to all of us collectively. In community laboratories and major research institutions, in regulatory offices and patient advocacy groups, in classrooms and kitchen table conversations around the world, people are wrestling with these questions together. And that collaborative spirit, that recognition that CRISPR's future should be shaped by all of us, not just the scientists who perfect it, gives me tremendous hope for how this story will unfold. Tonight, as you drift towards sleep, millions of your cells are quietly going about their ancient work. They're reading the genetic instructions written in your DNA, manufacturing the proteins that keep you alive, and repairing the inevitable small damages that accumulate during each day of living. It's a process so elegant and reliable that you never have to think about it until something goes wrong. But now, for the first time in the 4 billion years since life began on Earth, we have the ability to step into that cellular conversation and make careful, precise edits to the instructions themselves. We can correct the typos that cause suffering, add new chapters that provide protection against disease, and perhaps even enhance the capabilities that define human potential. This power comes with responsibilities that previous generations never had to face. We're becoming conscious participants in evolution itself, editors of the book of life. The changes we make today could echo through countless generations, shaping the biological destiny of our species and our planet. Yet there's something profoundly beautiful about this moment of transition. For billions of years, genetic change happened through the random dance of mutation and natural selection, a slow, undirected process that created wonders but also allowed tremendous suffering. Now we have the opportunity to guide that process with intention, compassion, and wisdom. The bacterial immune system that inspired CRISPR worked through trial and error across geological timescales. We don't have that luxury of deep time, but we do have something perhaps even more powerful, the ability to learn from each other, to share knowledge across cultures and continents, and to make collective decisions about how these tools should be used. As this gentle exploration comes to its close, I'm reminded that science at its best is not about dominating nature, but about understanding it so deeply that we can work in harmony with its patterns and processes. CRISPR doesn't force genetic changes through brute power. It works by speaking the same molecular language that cells have used for billions of years. It's a conversation, not a conquest. And perhaps that's the most important lesson from tonight's journey. As we stand on the threshold of an age when genetic modification becomes routine, we must remember to approach this power with humility, wisdom, and deep respect for the complexity and beauty of life itself. We must listen carefully to diverse voices and perspectives, proceed thoughtfully rather than hastily, and always keep human flourishing in all its wonderful diversity at the center of our efforts. The story of CRISPR is still being written, and you are part of that story. Whether through the personal choices you make about genetic testing and therapy, the conversations you have with friends and family about these technologies, or simply the values you bring to these discussions, you help shape how this remarkable tool will be integrated into human society. So sleep peacefully tonight, knowing that while these questions are complex and the stakes are high, humanity has shown throughout history a remarkable capacity for rising to meet great challenges with wisdom and compassion. The same species that learned to harness fire, develop agriculture, and cure infectious diseases is now learning to read and edit the fundamental code of life itself. And in laboratories around the world, as you rest, dedicated researchers continue their patient work, asking careful questions, conducting rigorous experiments, and gradually expanding our understanding of how genetic modification can be used safely and effectively to reduce suffering and expand human potential. The bacterial immune system that gave us CRISPR operated in darkness for billions of years, unknown and unappreciated until curious minds finally decoded its elegant mechanisms. Now, Illuminated by scientific understanding and guided by human wisdom, it serves as our partner in the ancient art of healing. Tomorrow, when you wake refreshed and renewed, you'll do so in a world where genetic diseases that once seemed incurable are yielding to precise molecular interventions, where crops are being engineered to survive climate change and feed growing populations, where researchers are exploring whether the aging process itself might one day become editable. It's a world full of both tremendous promise and significant responsibility, a world where the boundary between natural and artificial becomes increasingly fluid, where the line between treatment and enhancement continues to blur, and where the very definition of human nature is quietly evolving. But it's also a world where scientific wonder continues to unfold at the pace of gentle discovery, where each breakthrough builds patiently on the work of countless others, and where the greatest technologies emerge not from hubris, but from humble curiosity about the way things work. Sweet dreams, gentle listener, and may your rest be filled with the quiet wonder of a universe where life itself has learned to write its own revisions. The bacterial scribes that first discovered CRISPR billions of years ago never could have imagined that their simple survival strategy would one day become humanity's most elegant tool for rewriting the stories written in our cells. As you settle into sleep, your body continues its nightly symphony of repair and renewal, each breath drawing in the same oxygen that has sustained life for eons, each heartbeat pumping blood through vessels that carry the same fundamental building blocks that connected our earliest ancestors to this very moment. And now, for the first time since life began its long experiment on this small blue planet, we have the ability to consciously participate in that ancient conversation. The journey from bacterial immunity to human healing reminds us that the most profound discoveries often come not from grand ambitions, but from patient attention to the small mysteries that surround us. Someone noticed repetitive sequences in bacterial DNA and wondered what they might be for. Someone else observed that certain bacterial strains survived viral attacks that destroyed their neighbors. Each question led to another, each observation to a deeper understanding, until suddenly we found ourselves holding the keys to the genetic library of life itself. If you found comfort in this gentle exploration of CRISPR and the future it's quietly writing, I'd be ever so grateful if you shared this journey with others who might benefit from a peaceful dive into the wonders of science. A like, a subscribe, or simply sharing this video helps create a community of curious souls who find solace in understanding the elegant mechanisms that govern our world. And wherever you are tonight, whether you're drifting off in Dublin, finding quiet in Tokyo, or settling in for the evening anywhere else our sleepy science reaches, know that you're part of this extraordinary moment in human history. We're all temporary custodians of genetic information that stretches back to the very origins of life, and now, perhaps for the first time, we have the wisdom and tools to tend that inheritance with both precision and care. Until we meet again for another gentle journey through the landscapes of scientific wonder, rest well, dream peacefully, and remember that even as you sleep, the ancient molecular machinery of life continues its patient work, reading, writing, and editing the stories that make you uniquely, wonderfully, irreplaceably you. Good night.